And we will be doing the usual Q&A. We've been gathering the online questions. Feel free to keep sending those in. The microphones will be on, but since we have some folks joining us, I thought we'd give them a chance to share their thoughts on the topics that were covered this afternoon. So let's, Wang, let's start off with you. If you want to go ahead and share a little bit about your insights about what you heard discussed today.
Yes, yeah, my name is Jani Wall. I'm on the new drug side, Division of Pharmacometrics under the Office of Clinical Pharmacology. Actually, our division is a big proponent of this methodology. I guess even before I joined FDA in 2003, there was already a dedicated group, even though back then it was not a division. They were the pioneers to, I guess, promote the application of model-based or model-informed analysis, specifically on the new drug side. Over the last, I guess, 16 years after I joined FDA, we grew from a small group of like 4 or 5 people to a division of 30 people today. And at the same time, we have seen the application of model-informed analysis across the entire, I guess, drug development, from the first-in-human dose selection all the way to even post-marketing dose optimization after the drug is approved. And I'm glad, yesterday, you have seen the application of model-based analysis in the generic drug side as well. In fact, when I was working on the new drug side, we had a lot of collaborations. For example, the dose scaling approach, and the last example, the long injecting. Actually, that was when I was a team leader, and I worked with my current deputy when he was the primary pharmacometric reviewer. We reviewed the 1 month injection formulation and worked with the company Johnson & Johnson. I think for the first time we accepted a regimen that was not even studied in the clinical trial, based on the model-based analysis, and we put a lot of model-based recommendations in the product label. For example, what if you miss a dose because it's a once a month? In fact, the first dose, the second dose was after 1 week, and the next dose is once a month. So the sponsor came up multiple realistic scenarios. What if you miss the dosing window that was studied in the trial? How large that window should be, and what should be the re-titration schedule? And all this, I would say, I guess I'm going to use Liang's new terminology. Model integrated evidence was used to support the label language, and they were all included in the final product label. And those were the early successful stories. Even up to like today, I still use that example to encourage, the sponsors, whether it's generic or new drug, to come up with novel approaches. Whether you want to propose a optimized dosing regimen in the new drug side, even though it was not studied, but you believe after analyzing the phase 3 data, you believe this new derived regimen or model integrated evidence support a better optimized regimen, the sponsor should propose that, provide justification. And then, of course, we will review and evaluate, and who knows, you may end up with a so-called derived regimen that was never studied in the trial. So I want to congratulate the workshop and of course our Office of Generic Drug side on the. You have seen some areas actually that's even more advanced compared to the new drug side. For example, the inhaled product, where not only the traditional PBPK or PKPD, but the dynamic side on the formulation side was the chemical engineering model was introduced. In fact, on that area, the new drug side is still learning because, again, it's also highly relevant for the formulation development. So we will learn from each other, and I guess we'll help each other with all the good examples. Thank you.
All right. Thank you for those insights. And Murray, you're new to the panel. Please follow up based on what was discussed this afternoon. Give us your insights and background.
Okay, well, thank you very much. Well, good afternoon, everyone. First of all, thank you for inviting me to be on this panel. It's really an honor to be here with you guys. I have to say that, model-based approaches have always been my baby, almost, since I've been doing it for like 30 years. And it's really refreshing to see how important it is now for the Office of Generic Drugs. The Office of Generic Drugs was the first one to come up with a guidance that was based on modeling. That was the corticosteroid guidance, so in 1995. So that was really ahead of its time. And then there was the dose scale analysis that was proposed in the late 90s, so that, again, model-based. So this was exciting, but at the same time, when you were talking about model-based stuff with people at OGD, they would say, we don't accept model-based approaches. Yes, you do, because you have the corticosteroids and you have the dose scale, but no, we don't. And they were very close, and so there was a kind of a weird, weird thing. And so now, when I see the change that has occurred in the last, I guess, 5-10 years, by this new leadership that is at OGD. It's really refreshing. It's really nice to see. So thank you, Dr. Zhao. I can see a big difference. Dr. Lionberger, since you're there at OGD, a lot of good things. So I'm so pleased. What I would propose, though, is to be careful sometimes, and also to be consistent. Sometimes I think OGD is very, very demanding, and sometimes you wonder, there's some places where the demand is really much more than you think would be needed. So, for example, we talked about orally inhaled products. Me and others have been saying for like 20 years that you see everything that you need to see on the PK side. But, OGD has always been very demanding of this clinical study, which is very difficult to do, and at the end is not very discriminative. But then, on the other hand, sometimes we're less demanding. And so I was a little bit puzzled by the presentation on the PBPK, which when I saw the predictions and the fitting, it really looked awful to me. So I couldn't understand how this could waive a clinical study. That doesn't make any sense to me, because if you waive a clinical study and, understanding that the clinical study is needed, then the model really needs to be validated, needs to be good, needs to be solid. If the clinical study is not needed, then that's another issue. Then it's supportive, but so I'm just wondering here. Sometimes we're very demanding in one part, and maybe we're not in other parts. So I don't know. But overall, I don't want to be critical. What OGD has done is tremendous. The presentation this afternoon were great. So thank you, and thank you for doing everything that you're doing.
All right, and thank you for those thoughts. We'll continue that discussion and things going on, and I know that Liang, you led off this whole session on quantitative methods and modeling informed decision making, and Robert, you're going to be wrapping up at the end of the day with thoughts on everything. What I thought as we do is to continue this discussion is back up a little bit to check in on the questions we had from some of the earlier discussions, the presentations we had during this session. We do still have the presenters here in the room, and we can get a mic to them if you've got questions specifically to anything you heard, or if anything you've heard from the panel so far, you've got any thoughts or questions, this is where we're going to open it up for your input. So just like our other panels, if you've got questions, come on up to the microphone. Otherwise, we'll go ahead and start with our or any of you will ask questions.
So what questions I'd like you to ask us.
Oh, excellent.
Get to how do you integrate the model-based approaches, both on the quantitative clinical pharmacology and the PPK, into your development programs for complex generics. So really welcome discussion about that. What are they useful for? How do I fit them into the things that I'm doing in all the different therapeutic areas we've heard today? So we welcome any kind of questions or discussion around that as well.
All right. And so if any of you out there have thoughts that you'd like to share based on Robert's question, please come on up. But since we've got somebody at the microphone, the floor is yours. Please help yourself.
Thank you. I actually have a question for someone not up there, but was one of our last presentations given by Dr. Gang. It was on Emacs modeling and imputation. And just a clarification on one slide, there was a slide with some box and whisker plots, top and bottom, compared two different scenarios. One scenario, I think, was imputation at maximum concentration. I didn't quite get what the other scenario was, so maybe you could just clarify. I know you were short of time and you had to run through it, but maybe you could just clarify what those two plots represented.
Yes, so
thank you for that.
Thanks for the question. So the top is the result without the imputation. And the slides here in the lower group is the one with the imputation. So the point of the plot is showing, because for that particular case, maximum methacholine concentration were imputed for both T arm and T and R arm. So they're manually changed to make the TR differences smaller. So that's the point of that presentation. So that imputation is to impute the data. Yeah, remove, because these values are on null value. They're unmeasurable.
Okay. And again, any of the discussion you want to have around this topic that we covered, all the presentations, please come on up to the microphone.
Sorry, I just want to continue the question about imputation. And you seem to criticize the way to impute the maximum methylcholine concentration and understand where you come from. And so if the maximum methylcholine concentration is that right imputation approach, what imputation approach would you recommend?
I couldn't say there is one standard. There were cases where there are only small, very small portion, I would say about 3%, that needs to be imputed. And in those cases, actually the firm are recommended to exclude those data, and they followed it. And that's the approach for that. That's one way to handle that.
Sorry, I didn't quite get you. You mean the 3%?
I mean, if it's small, if it's only small portion of missing data,
yes.
And one way to go is to exclude those.
What about the 10% or 15%, which is
10%, 15%. There is no standard answer, and so that's why we put our recommendation is if you think your approach of imputation is valid, you could provide justification to say, oh, if I pick this imputation method and it won't cause biased estimation of F. Yeah.
Thank you.
Well, we
question.
are collecting questions. I'll use this time gap to address Murray's question. You know, I think you, thank you for your compliment, first of all. So you mentioned that for the PBPK modeling, you're not quite getting into the agreement that that can be sufficient to support regulatory decision making. So my address that is, the modeling practice. I hope all the audience has an appreciation of a different flavor when we're suddenly talking about the modeling. Things suddenly become complex, and as compared to other type of regulations, so there's lack of standardization. We should have an agreement on that. So it's not like a less than 5% or bigger than 5% based on the experimental data. It's meant an integration of knowledge, of understanding, and there will be an engagement of a difference in opinions. The case, yes, it is groundbreaking. It's a kind of first case that we used PBPK simulated result to assess the risk of post-drug approval. The fitting, I agree, may not be perfect. The decision is made not only based on the whether the fitting is good or not. It's based on a totality of understanding of the mechanism of action and the literature report. Also, the PBPK platform has been validated across a series of formulations, like a cream, gel lotion, also with chemical entities, with similar degree of measures. So all the platform has been, I'm using like EMA terminology, has been qualified that way. So we also, the structure of the model has been verified. And we validate the model against the literature data. We also assessed the risk. What if the model goes wrong? So all the things has been put into consideration. I think given the time limit, I don't think we have put all the details out. In that regard, I think Altheria is writing a review article. I think anything new put in the field, groundbreaking, we anticipate we anticipate difference in opinion. We really welcome difference in opinion. That way we can, we make everything public, transparent, and that way we can further advance the field. That's really appreciate the feedback that given by Ray. That's kind of my short response to that question. I don't know whether if you want to follow up.
Just want to make a point that I'm not against PBPK. I think PBPK it's important and it continues to be developed. It's going to be very important in the future, if it's not even important now, because as we know, for waiving of drug-drug interactions, it's very useful. What I'm saying is that the guidance that Europe has come up makes a difference between what you're using the PBPK for, and so you have A if it's only supportive, or B if it's in the middle, or C if it waives a clinical study. And so if it waives a clinical study, then it has to be validated. The validation demands are much greater. Now I don't know about this. All I know is what you've presented today. Okay, so I'm looking forward to see the paper when you publish it, and maybe what we saw was not really reflective of the rest. Okay, so I don't know. Okay, but what I see is that it's waiving a clinical study, so therefore it should be perfect. Okay, if it's supportive, it's another thing. Okay, and again, we've talked. This is for diclofenac, right? Yes. I mean, anyway, it's just that things have to be consistent.
I think we do need to download more details about this application,
sure.
including the package does include the PKB data, which shows the two products really have overlapping PK profiles.
Maybe if I can also make another comment, that it's separate, but when we say that, when FDA says that you can use a nonlinear mixed effect model, a modeling approach, I think that may not be precise enough. I know that FDA doesn't want to and can't endorse one software or one tool versus another. But, if we just look at the two main nonlinear mixed effect modeling approaches, an EM approach or FOCE, I mean, they're very different. And, one time you're going to use one, you're not going to use the other. The results that you're going to get from them are going to be different if you have, data that is missing, etc. So I would prefer that in the guidances, maybe OGD be a little bit more, maybe say which one we should use. I mean, because it's very different. I mean, I personally, as a modeler, I know which one to use when, okay? But someone that reads the guidance wouldn't know.
Yes, thank you for that. We hear you in the guidance development process. We definitely need to build clarity and also put ourselves into the applicant sponsor's shoes. Trying to give kind of a type of standard when to what do we expect and to what extent and what can be used for what. When we talk about a model, we always talk about model for purpose. It depends on the degree of validation also depends on the utility, the purpose. Even for the model verification validation also depends on the purpose and the risk. So we need to put all that into account.
Yeah, I mean, to your question on the nonlinear mixed effects model, I think the field in some sense is a little bit to blame on that because of the tight linkage between algorithms and implementations, right? That it's not easy to say, take this algorithm and go use your favorite implementation. It's like there's much more, so I think the field has to be sort of more established in this area, has to sort of separate the implementation from the algorithm so that you can determine is that what's the best algorithm. And then FDA, because I think we're reluctant to say, well, use this software, right? Which should be a sense of an implementation rather than an algorithm decision, scientific decision about what the appropriate algorithm is for a particular type of problem. And I think that's an area where I think we could do some work with the community on how do you sort of specify what we would really find acceptable.
Okay, we had somebody get up in the room with a thought or a question.
Thanks. I didn't want these guys to have all the fun, so I wanted to jump in. This is another question for another presenter earlier. This is on CFD and really the credibility or validation of the CFD. I took some interest in scintigraphy, and historically scintigraphy's not been sufficient for primary data for establishing what's going on locally in the lung because you're changing the RLD itself. But I'm wondering, and maybe you can just address this question, is scintigraphy itself sufficient for validating a CFD model in a specific product? So let's say I have meter dose inhaler X. I run a scintigraphy study along with CFD to validate the CFD for that particular product. Is that something that could be found sufficient for credibility? And sorry if I messed some of that wording up. But.
Sure, yeah, thank you for your question. So for a CFD model, sure, I think it could be useful information for validation. I think you make a good point that if you're using scintigraphy, you are modifying the formulation. Potentially, you could even have a CFD model that considers that formulation difference. That might be hard to do, but if you could do that, that would certainly be the ideal case where you know that you're validating against the actual case of interest. But yes, I think that there's a big difference between saying, oh, I'm just going to use scintigraphy by itself as a stand-in for clinical study, PD or clinical endpoint study, versus if you're supporting your CFD model. Now that said, if you only have a CFD model and you're trying to waive your clinical endpoint or PD study, going back to context of use, which I was describing, it's still probably a very high-risk situation. You're probably much better served if you're not just using CFD or using some other in vitro approaches or some other data from somewhere else to show that you've proven your point, basically.
All right, and let's not forget about the online folks. Ray, have we got any input, any comments, questions from the online folks as well?
We do have an online question. What's the difference between PD study versus clinical endpoint study? I thought they both give the same information.
I think there's a fuzziness in the boundary between what we call a PD study and a clinical endpoint study. Sometimes Yanning can talk about in new drugs, sometimes there's cases where things that look awful like a PD endpoint are the primary clinical outcome of the study. But certainly there's other cases where they are very different. So there's a fuzziness to that boundary there. Like I would say, I think one area where something we talked a lot about today were the inhalation products and the FEV1 measurements, right? Which are, certainly a pharmacodynamic response to the drug, but in many of the pivotal studies, those are the endpoint there is sometimes the FEV1 in the clinical trial. So that can sometimes be both. Instead of looking at, is your asthma under control, you look at a quantitative measure. So, there's a fuzziness in the boundary between there. I think generally in the Office of Generic Drugs for Bioequivalence and our regulations talk about favoring pharmacodynamic endpoints before clinical endpoints because we think that, basically because of the sensitivity to the formulation changes in the drug exposure that we generally think the pharmacodynamic endpoint might be more sensitive to the clinical endpoint because a clinical endpoint in many cases may be further downstream than the sort of measurable pharmacodynamic effect. But Yanning, maybe you wanted to comment a little bit on that, on the nomenclature language there.
Yeah, again, I think on the new drug side, there's a clear difference between PD endpoint trial versus clinical endpoint trial because whenever we call a pharmacodynamic endpoint, which automatically implies it's not a clinical endpoint trial because on the new drug side, the key evidence or the primary evidence is based on clinical endpoints. If there's a one endpoint, something like viral load or even HbA1c or the PSI, all these are, you can view them as a pharmacodynamic endpoint, but they are validated to an extent of surrogate endpoint. So in some sense, they are treated as almost like a clinical endpoint to have a full approval. In fact, probably it's easier to see the difference between a PD endpoint and clinical endpoint on the biosimilar drug development side. Because there, again, there's a clear separation between a phase 3 trial that's based on a clinical endpoint to show similarity between, let's see, the reference product and the biosimilar product versus a pharmacodynamic endpoint-based trial to show similarity between those two products. Because, again, most of the so-called phase 3 similarity trial is still based on clinical endpoint, even though I would say there's a trend to move towards a more PD endpoint-based clinical trial. Therefore, over there, you can clearly tell the difference. One is a PD endpoint-based, still it's a clinical trial, but endpoint is, if it's called a PD endpoint, that means it's not the clinical endpoint, maybe somewhere on the causal chain between how the drug works and ultimately the clinical endpoint. But there's a reason to pick a certain PD endpoint because it's more sensitive, it's less noisy, and therefore maybe a smaller sample size, a shorter trial. So there is a clear difference.
Okay. And I think microphone number 2 came up a little bit before microphone number 1, so go ahead, whatever you wanted to share or questions you might have.
Sure. Good evening. Thank you very much for last 2 days support in simplifying the complex product development of these generic products and support of SBIA. And these are really helpful for us. And so one of the question, which is a complex API source is coming from the natural source with the 10 different active moieties in it or more. So when a product like that have a systemic application, which is either oral or topical or injectable product, in that case, obviously pharmacokinetics is not easy for us to come up with the equivalency because it's more than one moiety. And we believe that its clinical endpoint is the best solution to show the equivalency between the test and reference product. in that situation, if you agree that one and any other modeling would help to avoid the clinical endpoint study, or still we need to stay with the clinical endpoint is the only option in such kind of the product?
There's always options to a clinical endpoint study because we have our pharmaceutical science and physiology understanding of the product. And as we improve that, we eventually we'll be able to understand what's the most efficient thing to measure other than the clinical endpoint. So certainly there's always the potential for alternatives. I would say for complex mixtures that are intended to be systemically absorbed, FDA's, even our general guidance for many years has sort of indicated that you can select appropriate components to measure in the PK product. We don't say that you have to measure every component that's in the product in your PK study. And I think that's a good application where maybe the modeling and simulation from a molecular level might help you to really understand what's the absorption of every one of these components and which one is going to be the most sensitive and appropriate marker. And it could be one or two. So I think there's a lot of opportunities there for the sort of mechanistic, even out of the chemical level, modeling of what the difference between those different components might be in their absorption and distribution, to say, what's the one that's going to measure that's going to tell me what are the characteristics of the mixture as a whole? And so there might be approaches like that to be more quantitative about that decision. But certainly in the past, FDA sort of said that's our approach that we would be in favor for systemically absorbed complex mixtures, is to identify a particular component or components for measurement in the PK study.
You are also welcome to, in addition, just follow what Rob has mentioned, to assess the API sameness. If the mixture, there may be new methodology needed to assess the substance sameness. In that regard, it's not only FDA's interest, but also the interest of the firms to propose new methods and how to evaluate sameness.
Thanks.
Okay, microphone number 1, please.
Thank you. This will be my last question. I started my industry career 20 years ago in the UK. Then within 5 years' time, we were able to develop a number of generic inhalation products in the UK. Over the past 20 years, actually, most of inhalation products have a generic product on the European market. And now look at us. I saw a slide this morning. So many inhalation products. Only we have one generic DPI approved in the US. Now my question to you guys, can we learn from European colleagues' experience? Because in any sense, we should do differently here so that we can increase access to the cheaper, high-quality generic products in the US.
I mean, I would say I think you got the point about what is high quality for the inhalation products. If you have an environment in Europe where you're not, in the past, haven't been, you heard a lot during this workshop about device comparison and patient use aspects, right? If you take those, that's part of the US system and expectation that maybe not the case or expectation in other regulatory systems about substitutability and at the point of pharmacy dispensing, is why in the US system we're so concerned about patient use, because we expect that once a generic product is approved, that it's substituted, without particular intervention or training. And if you have a different type of system that says, well, these are, in the US language, it would be pharmaceutical alternatives. Here's another option. In the US system, there's lots of options for inhalation products. It's just that we don't think that they're directly pharmaceutically substitutable because there's differences that aren't allowed in our system. So there are some differences in the system and substitutability. And I think we definitely have a large number of research activities looking at what's the really appropriate measure to ensure the same local delivery and drug delivery similarity between the products. And as you can see, some of the guidances are coming out show more efficient approaches, and We encourage, and many companies are coming in to meet with us to talk about alternative approaches in the inhalation area, and we encourage that in the future.
All right, let's take an online question, please.
How common has computational modeling been used by industry? Have many sponsors been using this method, and has the agency seen these models being used for all different products or only to the ones discussed in the presentations today?
For industry, so maybe that's a question people in the industry could answer better than us at FDA, because I think we only see things really when they come to the end, right? It's a question of what are companies doing internally. And, I understand the spirit of the question, right? What are other people doing? I think this is an area, and there's a lot of areas today where I think there are opportunities for industry, for the parts of the industry that are willing to be innovation drivers or thought leaders, right? So if you're, and I don't know what the spirit of the question was, and the person's not here, so I can't look in their eyes and determine what's in their soul. But if it's like, well, is that question coming from fear of being left out? Or it's, I don't want to be the first one to do this, but if other people are doing it, I'm okay with it. That's one perspective on how to work. But there's another perspective, which is, I want to leverage new tools to move into a leadership role where I can be the first person to do different things, or the first person to move into a more complicated generic competition with a different level of evidence. So I think there are opportunities for innovation to be leading opportunities. So I don't know that the question is, well, I'm not going to do this until other people are doing it. Certainly every company and business has to determine their strategy, but I think there's opportunities for leadership here. But I think maybe Leon can talk about some of the examples that we've seen that have reached us. I also say that for us, as users of this, as we have to make decisions about product-specific guidances and application review, we use these tools all the time, and they're extremely valuable for making decisions. This doesn't get into waive my study or things like that, but what should I do? These are things that we use all the time, and I think companies that use them, even if they don't actually say, well, I've got to put this in my ANDA, if you use that to say, I should do formulation A or formulation B because this one's going to pass the BE study and this one isn't, you can save your company and your development program significant resources by doing that. But that may never show up to us. So it's not necessarily the right question to say, well, what has FDA seen? It's really a question for industry. What are the value cases in industry where you've made better decisions?
Yeah, I can follow on with what Rob has been addressing. I think we use computational tools to benefit the applicant. So, every year we have a workshop to get a priority list for research what the area that make industry suffer. Then can we use some creative computational approaches to bridge the gap based on the current knowledge? It's really tackling on areas where industry, the firm has a problem. For example, we do see there's on top of the topical product, we do see applicants using CFD models for orally inhaled product. So for the modeling applications, unlike other type of regulations, it's a big picture. It's a kind of a built into the overall regulatory program. The return on investment can be substantial for complex generic drugs. For for conventional BE assessment approach, there may not be a value to use this quantitation, may not save much by using this computational approach. But for complex product, the return on investment could be humongous.
Okay. And Murray, I saw you reaching for the microphone. Please, your thoughts?
Yeah, I will just say one thing also related to that. There's the history part as well. I mean, for a long time, OGD was reluctant that we would bring any model-based approaches besides the corticosteroids, obviously, and the dose scale. So now it's a big change, and so the industry, in part, to answer the question, the industry is not used to that, and they're unsure where they stand. But I think the model-based approaches are really important for long-acting injectables. I mean, for long-acting injectables, the guidance right now, asking for steady state, and for those products that have such a long half-life, it's not really feasible to do generics using standard approaches. So model-based approaches are really the way to go for those ones. And so I think the industry will probably propose to you, things related to that.
All right. And I do see one more question here, but unfortunately we're out of panel time. This will be something that can be discussed later. But before we finish out the panel, I actually, there's many people throughout OGDs, SMEs, and thought leaders that have been working with Brenda and the SBIA team to make this event possible. And one of the strongest voices there, wisest minds, is Dr. Robert Lionberger. So we've asked him if he's got some closing thoughts on the full 2 days, the things that were discussed, to kind of round out the rest of the...
All right, thanks. Thanks, Jeff. So it's been a great event. I've heard from our SBIA, that our peak attendance was something like 2,200 people online listening into this. So there's a wide interest in this. And an event like this, it takes a lot of work. So I really want to thank all of the people within FDA, so Brenda and her colleagues at SBIA who do the organizational work. I'd like to thank the speakers from OGD and OPQ who brought their scientific expertise to here. I'd like to thank Karen Bankston, our project manager, who helped herd all of these things, chase down people's presentations and all the things. So thank you, Karen, for that work. Let's give her a round of applause. And really want to thank all of the attendees for your interest in this. And I think that's a great testimony to the health and the future state of the generic industry, that so many people are interested in what's going on at the leading scientific edge of this area. And the complex generic products, they're important. They're important both to the patients that are looking for generic competition in these areas, and they're important to the generic industry that's looking for opportunities to supply that and develop new products and new business areas for themselves. So there's a lot of interest and value that gets created by this. And from FDA's perspective, we hope that this type of meeting really helps those synergies grow. It helps companies see more clearly, how do I get in and supply products in these more complex areas? And what we've tried to illustrate in this meeting is sort of the progression from the science and research that we do that's supported through the generic industry user fee program. So the industry is investing in this in a way that we hope has helped building the future success of the generic program. That these research investigations into new analytical methods for the complex products, into the CFD methods and how you use them for generic products, are building out the foundation for the approaches to these complex products. And so here at this workshop, we started off with a discussion of our pre-ANDA meeting process. This is new in our second iteration of our user fee program. I think it's very successful. We're receiving, around 100 meeting requests each year through this program. Each one of these is really on point to something that was touched upon here, a new approach, a complicated question of how do you do something more efficiently. So we think that that's something that really, again, as I said, be a company that's driving the innovation that says, I'm looking for a new and better way to do this, and willing to sort of come and talk to FDA about this, leveraging the scientific advances to do these new approaches. You also heard a lot in this workshop from our review staff who review these applications once we get in here. Because again, as we said, the goal of this is to make the complex products accessible to the American public. So it's really important that we, I would say, build the plane with the science, but land the plane to deliver that product through the system. And I think this can be very helpful. A lot of these aspects of the complex products, they reflect the complexity of our regulatory system. So the more that we can be straightforward and clear about what those requirements are, You know, all of the new things about drug device combinations that you need to be aware of. Those are important. You know, they may not be the most scientifically challenging thing, but if you have a complex product and you want to deliver it to the market, you got to get things like your syringe right. You've got to get your pH adjuster right. The sort of basic level things as well as the complicated questions. So we hope that this workshop has helped really clarify in these complex product categories the key things that you have to be aware of as you're moving a product through development and through the review process. So I hope this meeting has been very valuable to you. It's exciting for us at FDA and OGD to do this and to organize this and to keep these things moving forward. And we look forward to doing it again next year and hearing about new things that are going on both in our review process and our scientific advances in these areas. So with that, I'd like to close the meeting and wish everyone who's here in person, and encourage more of you to come in person in the future, safe travels home, and thank you all online for participating and paying attention to these important topics. So thank you all very much.